Get ready to get started here. I want to introduce Dr. Garner. When I was appointed the chair here, I have to tell you this, the first call I got was from Skip Garner. He wanted to know something about me and what plans were for the department, what was going on, and he wanted to offer some of his help and advice. And I really do appreciate that. I think though he was just trying to check up on me, make sure I was quality, but hopefully he still thinks so. Dr. Garner is also a member of our development board, which will be meeting later today. And once again, the development board is people who advise and give help to the department. But I also want to highlight some of his credentials. Certainly, he's our graduate. Let's see, that says 1976. That can't be right.
That's pretty ancient.
1976, got a BS here, went on to Wisconsin-Madison to get a PhD in plasma physics. And then, as he's going to be talking about, some strange things happened. He went to work for General Atomics in fusion, did a lot of things there, and then he somehow got involved in the medical school. And so then went on to Southwestern Medical Center at University of Texas, chair, professor, distinguished name chair there, and went on to do quite a bit more there. Then went to Virginia Tech and was involved in the bioinformatics and medical informatics section as executive director. And I'm just zooming through, highlighting all these things. Research associate dean for biomedical affairs. That was at BCOM in South Carolina. And now kind of semi-retired and a consultant for a lot of medical schools and organizations. So I just also wanted to highlight though that he and his wife rescue dogs, horses, and birds on their farm in North Carolina. So I think that's pretty cool. And with that, I'm going to let Skip go ahead and present.
Sure. Well, first of all, thanks for inviting me, and it's great to be here. And you passed, by the way, the very first interview, so you know, it works both ways. You know, normally if the professors are giving me a grade, it's great to get back. So it's great to be back in Rolla, and it's great to see a large number of nuclear engineering students, because I think that that's a critical part of our future. You know, you've all heard about, you know, the carbon problem and global warming, and so this is one of the solutions. And so it's great. Go with it. Okay, so I would like to tell you a little bit about medicine, because I've also heard that, you know, there's going to be a new biomedical engineering department here, and there's the medical physics, you know, nuclear radiation, nuclear effort to get something more going there. And since I've been at medical schools now for, I don't know, 40 years or something like that, and I'll give you a flavor for the kinds of things that happen there, mainly through what my research effort is right now, and what you could expect to hear and see when you start having those kinds of activities here. All right, so with that, let me just start off by saying one of the first things you do in all medical talks is you talk about your potential conflicts of interest. Above the line are my companies, Iridia I kind of advise for. That's one of those DNA data storage companies out in California. I'm going to talk to you a little bit about Orbit Genomics, because that's the cancer diagnostic stuff I'm doing right now. And I've consulted for those companies down there. I really wish I owned one of those. It would have been far better, and this meeting would have been at my place in the Caribbean. So with that, let me just tell you, I was at General Atomics, and I was in the first 6 years. I spent most of my time in Japan, a little bit in the Soviet Union, France, Germany, England, working on fusion stuff. The second 6 years, I was in an internal think tank there called the Institute, and we worked on a bunch of stuff. I'm going to show you some examples at the end. But one of the primary things was I read this little article in Scientific American, and this was in the mid 80s that said, mid-late 80s, that said, we're going to have this thing called the Human Genome Project. We're going to sequence our genome, figure out what all the DNA means, and we're going to cure cancer. And so I went to my boss at General Atomics and said, hey, I think we should work on some of this biology stuff, right? And he goes, okay. And, but he goes, is there any money in it? And I said, there's a $3 billion coming down, from Congress next year. And he goes, all right, okay, get on it. So I go across the street to the Salk Institute. General Atomics is at the corner of UCSD, Salk Institute, and Scripps Medical and Scripps Oceanography. You're from San Diego, you know that. And so I went there, I met a guy who's an MD, PhD. We started working together. And so what happened was we submitted an application for one of these first human genome centers and won it. At the same time, University of Texas recruited he and I together because they said, we want the human genome to be here. So we moved to Dallas. They gave us an offer. 1 month later, we called up and said, hey, and the dean goes, there's not a problem, you're still coming, right? And then we go, yeah. I said, yeah, but we have this extra $40 billion. And he goes, all right, you get extra floors on the new building. And so we went there, and that's how we got started doing the Human Genome Project. We sequenced chromosome 11 and 15 the hard way the first time, just rooms and rooms of sequencers with technicians working 24/7, 365 for several years. Now I can do that in my lab at my company. I can do a whole human genome in 3 days. Well, I can do one genome per day with one little machine. It takes 3 days to go through, so I just have to stage it. But nonetheless, now back then, basically they spent $3 billion to do the first human genome. So that's the first one. I'm now doing genomes for less than $300 a pop. So that's what technology has done to revolutionize things. Portion of our genome, so there's 3 billion letters in, well, you're looking weird. So maybe you're 2.5 billion letters. I'm not really sure. But so that's fine. The average person has 3 billion letters, and in there, these are letters C, D, A, P, P, D, A, C, like that. There's a certain kind of sequence in there called repetitive elements or microsatellites. You see the word microsatellite. I became fascinated by these things in 1994. What they are is they're sequences that are like 18, 18, 18, 18, 18, 18, and they'll have maybe 20 copies of that. And so these are fundamentally different from the single base changes that happen in the fact that the DNA, when it's replicating, it gets better. So it'll add more letters or subtract letters and things like this. So they're much more variant. They have much more information in it. And so that's what I started working on in 1994 and publishing papers and working on stuff. And the rest of the talk about the diagnostics that we're building right now is based on that. So these are some of the other people's papers, where there's diseases like Fragile X, Huntington's disease, things like this that are caused by these type of repetitive elements. All right. And so the other thing you should know is all the forensics and paternity testing that you see on TV and that everybody's doing, you call up CSI and get somebody's thing, find out who's your mama, And so that's all done with microsatellite fingerprinting. These same kind of things are done with all that. It's the most used things. All right. So I have been researching these since '94 and have worked on various things. And a few years ago, launched a company called Orbit Genomics to create cancer diagnostics using these as the basic technology that operates these things. And so the idea is that since they're fundamentally different and they're more sensitive and they're more mutable than the rest of your DNA, they should be able to give you something that's more highly sensitive and highly specific to certain diseases, okay? Now, the other thing I noticed when I had an NIH grant for studying this thing was when the first human genome new technology called next-gen technologies coming out, people focused on calling in single base changes called SNPs. They didn't focus on this repetitive stuff. The technology was not focused on it. And so I went and got a bunch of sequence for some mother, father, daughters, and then compared them. And I saw that at these places, these repetitive regions, the accuracy of the sequence was only 20%. So 80% noise. Can you make a lot of discoveries when you have 80% noise? You know, you guys, what do you do in nuclear engineering? All you do is go, we need a hotter source, we need to take more data for a longer time. Well, I figured out that I could change the way the algorithms for how these things are simple. And so I drove the sequence accuracy up to 95%. That's a game changer. Now you can actually make discoveries. So we uniquely did that, and still other people don't know how to do this. So this actually belongs to our company. So all the discoveries within this portion of the genome, about 1 million of these places, we can do that no other people can do. And so given this and this technological advantage, we started making observations and have had a number of papers. We did this paper with Children's National in DC on medulloblastoma. Medulloblastoma, if you don't know, I'm sure you should know that because everybody knows. You know, they're tumors in infants in their brain. Okay, they're horrible things. They don't live very long. We studied that recently because it is in infants. These are people who've never been challenged by environmental insults like chemical exposures. And so it's really in their genes. That's the reason why they develop it. And we've published papers on breast cancer, lung cancer, liver cancer, all kinds of different cancers, right? And so using this technology, we've now started to develop some diagnostics where we compare a whole bunch of people who have cancer X to a whole bunch of healthy people, and we identify out of these 1 million loci that we now correctly assemble from using our algorithm, we identify those that can differentiate between healthy cancer, say lung cancer or something. And so we're doing that. And why are we doing diagnostics first? Why do you do diagnostics first in medicine before therapeutics?
Because it's a really bad idea to treat something you don't know what they have.
That's part of it. I'll give you another credit. It only takes about $100 million to bring a diagnostic to the market. It takes $2 billion to bring therapeutic to the market. So it's easier to get 100 million than 2 billion. Take my word on it. And so that's what I'm going to show you. Lisa, we can also do things like find these things that are diagnostic for what, like if you got a cancer, you're getting an aggressive cancer, you're getting one that's kind of going to slowly develop, and so you'll live for a long time. The other thing that we've done is there's something called companion diagnostics that now the FDA requires for all new therapeutics. Okay, if you get a therapeutic, what do you want to know when a doctor says, we have a drug we're going to put you on? What do you want to know personally about that drug? What will it do? What will it do? What are the side effects? Okay, I know you've got a few side effects. I can tell. No worries. I'm going to tell you. That's all right. Don't sue me. No, but the other thing was, will it work? The other thing is, will it kill you? Okay, a lot of times the drug that you get could kill you. All right, so now the FDA requires you to come up with a genetic test and say this drug was likely to work and is likely not to kill you. So we do that kind of stuff. And the last thing is new therapeutic targets. We find opportunities to attack a particular disease. Right now, we're working mainly on pancreatic cancer. Okay, why pancreatic cancer? It's a very rare cancer. There's only a few thousand people a year get that. But unfortunately, the time between diagnosis and when you die from pancreatic cancer is averages 4.2 months. So it's probably good to have, some therapies that could work on that, right? Okay. This is how we do it. Okay, read all this. There'll be an exam tomorrow. Basically, there's been a huge amount of money spent to sequence a lot of cancer patients and a few thousand normal people, okay, healthy people. And so, however, all that data, raw data is out there, but the places where microsatellites are all assembled wrong. They're inaccurate. We take it, so we've downloaded about 50,000 human genomes, and we use them for various studies. And we rebuild them at 1 million different places in each of those genomes. And so we database all that stuff. Then what we do is we go for the 1 million loci that are corrected, we go one by one and say, is the sequence different between the cancer patients and the normal people? And if it is, we go, keep that one, the rest of them we throw away. Typically, we find about 10, 20, 40, or 50 that differentiate the two different groups, right? So all this stuff is identify the places that are different, make certain it's not an artifact due to false discovery. So has anybody here studied false discovery and know what that is? Yeah, okay. You know what it is. So if you have enough data, you can find a correlation in anything. Exactly. So the whole point is you want to try and eliminate that as much as possible. So we do a lot of statistics in here. When we're done, we take these 20 or 30 things, we assemble them into one set and use them as a classifier to detect the difference between cancer and not cancer. And then what we do, this is called the custom target enrichment system. So what we do is then we take and we make a collection of a few hundred sequences that if I take DNA from you, I remove the DNA, and I only want to look at 30 or 40 different places. I use an enrichment kit to just extract that pieces of DNA and reject the rest, because I don't want to look at the full 3 billion, right? And so this allows me to look at it with more accuracy than, because it costs a lot to get 3 billion letters, but a little to get 115 times a couple hundred letters, okay? So we put it on our own custom target enrichment kit, which is a thing we order. And so you order, it comes in, you simply take and squirt your DNA into that. Then you extract it with magnetic beads, and then it's got the DNA you're interested in, and you throw the rest of it away. And so this goes into our clinical diagnostic test. So this is the thing that it analyzes all these important loci, and then we do validation on it, and then boom, we're ready to start clinical trials and actually analyze human genomes with that. Okay. We decided not to do pancreatic cancer first, because only a few thousand people a year die. We do lung cancer. How many people die from lung cancer a year in the United States? 175,000, something like that, 150, 175,000, depending on how you count them. That's a lot, okay? It's the #1 cancer killer by far. The next largest ones, breast cancer, etc. The next 4 are less than the #1, which is lung cancer, right? What else do we know about lung cancer? What's the 80% of lung cancer patients are smokers, or they're coal miners, or they're exposed to something like that. But mainly smokers. All right, 20% of them are not. But 20% of 175,000 is still large. But nonetheless, this is a cancer that you should have a really good diagnostic for, okay? Because people are dying, right? So what we did is we did the process I just showed you on a bunch of genomes, 500 of lung cancer patients and 390 matched kind of healthy people. And we found a set of 21 markers that then, when we use it to on the original set, it differentiates them. Okay, for those of you who know a bit about statistics, these two distributions are pretty well separated. A lot of times they're overlapping, but these are well separated. That's what you want, good separation, because that's when you get high sensitivity, high specificity for differentiating one thing from another. We then took these same 21 loci, and we put them on our own target enrichment kit and started sequencing new people who were healthy and those that were controlled. And this is the distribution we got. And so it still looks good, right? Still separates them. One of the big problems in medicine is that when you go to validate something, it doesn't validate. People write a lot of papers about stuff that don't validate. Well, our stuff validates. Look at the ROC, that's pretty darn good, right? And so this was the data we published in 2017. And went on right after that to actually found a company to do this. All right. So in essence, this is going to be just a little cartoon. But in essence, what we do is we have this component where we do discovery. We compare these repetitive sequences when you compare one thing to another. Those that respond to a drug, those that don't. Those that have cancer, those that do, etc. And what we do is when we have all the sequences, we extract DNA from a person, we run that DNA through our kit, put it onto a next-gen sequencer, and then it generates some sequence answer. We compare that to the thing that says you have cancer, you don't have cancer, and you generate a clinical report. So that's what we have. It's all running in Boulder, Colorado right now. And so we're receiving samples right now to do exactly this on patients. And we're starting, we're in a phase right now where we're doing clinical trials with Mount Sinai in New York City. So I want to change things here a bit. Let's see. Okay. Now, I thought for fun, what I would do is I had went and dug up our pitch deck we used a couple of years ago to get the money to start a company. So this, I just gave you the scientific background to a new cancer diagnostic. Now I want to give you the pitch that we gave 150 times, about 150 times, to venture capitalists and other people to talk them into giving us money. The company exists and we're in clinical trials. So what happened? We got money from one of them, right? Okay, so here's what we told them. I thought it'd be interesting.
How much money were you asking for?
We're asking for 5. We got 4.5. Yeah. So, well, 4.75. When the state of Colorado threw an extra $250,000 because we said we put the company there, and so they were doing it. Right now, we're trying to get a little bit more. Our next round will be 20. You got a good checkbook? Okay, well, then forget it. Okay. No, but it takes usually a couple of rounds. But one of the good things about this is in diagnostics is we're in clinical trials right now. So this diagnostic will be on the market 1 year from now, if we're successful. Okay. And so because we do so much research using existing genomes that we download from a public database, that's why we only needed $4.5 million instead of the usual 50 or 60 million, right? So that's why we also got money. That part. Okay, so I have a CEO, Dee Willis. I met her 20 years ago in one of my previous companies where I made a device that makes synthetic DNA. It made it 100 times cheaper than the existing devices and 100 times faster, etc. She had a company in Germany, in Greenville, North Carolina, and she approached me saying, can I buy some of these things? And I go, yeah. So I met her 20 years ago, and we sold her DNA synthesizers. And then time passed. I stayed in contact with her and other partners of hers. And then when I went to start this company, I go, I want her. Okay. And she started and succeeded in making several diagnostic companies since then. Okay. So this talk is usually given by her and I. We did it the worst possible time during COVID, right? But we did it all by Zoom. We've been Zooming all over the world all the time. And it's nothing like getting up at 3 o'clock in the morning and going, I'm going to pitch Singapore, at 3 o'clock in the morning. Okay, so it's her and I. This is the talk we gave. Okay. First, we want to introduce who. So we're the founding team. She's the CEO. I'm Chief Scientific Officer. This is our chairman of the board. And this is our lawyer. Okay. And you can see they're part of this thing called Ventec Partners. They're one of these companies that will come in and help you take your technology and make it into a new company. Okay. So they're kind of a consultants, but they also are attached to money and things like this. Okay. They have some history. you know, she ran parts and she made a company called Boulder Diagnostics. She worked for Sigma Aldrich and her, you know, you've heard of these companies before. This is the biggest chemical company in the world, I think. I'm not sure. I did a few things. I sold a couple of companies, one to Roche, another to somewhere else, to a UK company. And these guys have made other companies and sold them. And so this is the kind of people who you want to have as your leadership in the company. Okay, we got some advisors. We got the top genomics guy from Georgia Church, and he's at Harvard and MIT. This guy, John Min, is the best guy in the world for lung cancer. He's at UT Southwestern. He and I are old friends. We were together back then. We published a lot of papers together, almost all about microsatellites and cancer. And I was part of his comprehensive cancer center and things like that. We have a economics guy from Stanford and another guy who's a serial entrepreneur. His name, well, he's just super rich. But he didn't put any money into the deal. You know, you always want to run on somebody else's money, by the way. Okay, so we have this platform technology that will take blood or saliva and will analyze any complex disease, various cancers, neurological disorders, and heart disease. Those are almost those are diseases that kill most of us. And they're usually controlled by hundreds or thousands of genes, determines whether you've ever done. Our results are clinically actionable. You can do something with it. There's a lot of diagnostic tests that you can have that will tell you something, but can you do something to save someone's life, save money, or do something otherwise? Okay, our technology can be applied to diagnosing, treatment, or preventing some disease. We chose lung cancer because it's the #1 killer. It's the most preventable cancer. 80% of them are smokers. If you got everybody to stop, you would save 125,000 lives per year. Okay, 50% have some unknown carcinogenic exposure. And it's really, lung cancer is because of genetic mutations that you're born with or that you acquire over your life, and because of exposures to environmental factors, which also cause mutations that you acquire over your life. You just acquire more. By the way, you hear about this in nuclear all the time, right? You're exposed to radiation, you're going to get mutations and stuff like this. Everything that we're exposed to is going to affect us. By the way, we did a study once where we looked at radiation, O2 exposure. O2 is very caustic, you know. And we also did a couple of chemotherapeutics, all right? And we evaluated them for how mutable they were. How much did they mutate your genome? And we did this because sequencing, as I said, is really cheap now. So you just take a bunch of cells, you expose them to X-rays, you take some other cells and the same, you don't expose them. You let them live for a week or so, and you sequence both, and you find out which mutations are introduced. It's that simple now. It really is. So we published a paper, we did a bunch of those. By the way, one of the most intense chemicals to mutate your genome are chemotherapeutic therapies. Okay? All right, so what does that mean? It means the chemo that you get today will save your life today, but it's probably introducing mutations that will cause you cancers later on in life. By the way, you hear about that. Recurrence, your cancer is recurring, or a new cancer started somewhere else. It could indeed be the luck of the draw, but it could be because the chemotherapy itself is going to kill you. Okay, so cancer comes really because of those two things. They make some change in some cell that goes wild, and that becomes a tumor. Okay, lung cancer is the only major cancer without a routine screening test. Okay, now there is a screening consortium that is using low-dose CT to scan your upper body and to look for nodules or things in your lungs. It could be lung cancer. It could be something else too. It could be that. All right. This is depending on which site you go to get scanned and radiologist can read you. The accuracy for a radiologist to correctly call your lesion as to whether it's cancer or not is between 50 and 80%. Okay, really good ones are 80%, but that's kind of rare. Okay, so on average, 50% of the lesions that they think might be lung cancer turn out not to be lung cancer. Okay, so these are people who are screened. By the way, this is paid for by the US government, and they're paid for only for people who smoke more than 2 packs of cigarettes a day. Anybody here smoke 2 packs of cigarettes a day? Anybody here smoke a ton of new marijuana because you're in Missouri with the new law? Yeah, don't put your hand up. But nonetheless, we don't know about marijuana yet, so I'm not speaking about that. Okay, so they often need a biopsy to confirm, is it cancer, is it not cancer? And so there's this huge unmet need because a biopsy, by the way, costs between 10 and 10,000, 17,000, somewhere on average, depending on where you're at. And if something goes wrong, way more than that. Okay, but they got to go in and cut a little piece of your lung out. It's not good, you know. And so there's about 1 million biopsies a year from screening of lung cancer. About 500,000 of those say, you've got lung cancer, let's do something. 500,000 don't. They say, you don't have it. Okay, you just spent 500,000 biopsies, making a person miserable, having to spend 10, 17,000 per biopsy. By the way, there's about 1 or 2% of people die from a biopsy. Just getting biopsy because they accidentally make something and something happens, right? Okay, so the only way to verify from some CT screening whether you have lung cancer or not is to do a biopsy. Have those biopsies prove you don't have lung cancer. Wouldn't it be great to have a diagnostic test that takes a little bit of blood from you and has an accuracy, our accuracies are 92, 97% sensitivity and specificity, to couple with the about way less than 50% specificity of an LC, you know, computed tomography, you could avoid 500,000 unnecessary biopsies if you have a test that says that nodule is likely lung cancer because your genetics says you're likely to have lung cancer. All right, that's what our test does. You can do the math, right? 10,000 times a 500,000, and suddenly you can save about $5 billion in your savings if you have a test like the one we're developing, which is specifically to replace biopsies. You'll give it a little bit of blood, and it goes, you're at high risk of lung cancer. That probably is lung cancer. Then go ahead and do the biopsy. The people who say we're at low risk from our test, then you go, now let's wait a year and see if that nodule gets bigger or changed. You can afford to wait because it's better to wait because you're going to have 1 or 2% chance of dying just from the test, right? Biopsy. So you wait. All right. So the market for a test like this is about 3 to 5 billion in 2024. Okay. This is what our company wanted money for, so that we could address this market and uniquely capture, the funds. Venture capitalists, they care a little bit about the fact you're going to save some lives, right? By not them dying from it. What they really care about is, hey, if I give you $4.5 million, are you going to give me $40 million back in 5 years? That's what we're doing. Okay. So you have to have a really good test. So therefore, in conclusion, our company doesn't look at these single base mutations because we found them to be not really informative. We look at this unique thing of these repetitive elements, and we can find things that other people can't find. And that's why we've been able to make a diagnostic that other people could never figure out what we figure out. Okay. So we call ourselves a microsatellite company. There's about 1 billion of these repetitive elements. They mutate 10,000 times faster than in other places in your genome, and they reflect the current health of cells in your body more accurately than anything else, like I just said. So this is the pitch we gave 150 times and finally got money. Okay. This is some stuff I've already talked to you about. Same kind of stuff. Okay. Who here would invest money in this company? If you had money. Okay, let's see. All right, I gotta qualify it. All right, so my point is that now I wanted to expose you to this because now you've seen the underlying technology. But if anybody here is interested in inventing something and then making it into a company, this is what you suddenly have to become a business person. You have to become an actor. You know, you gotta talk to these people like you do. You gotta go and say, hey, Governor, another $1 million, we could make, you can even make him into a nuclear engineer. So, you know, God, I know I'm gonna get sued here. This is all for, okay. All right, so what I wanted to do there was just simply to illustrate that depending on which one you go in your life, there's lots of other acumen that you may have to build into your education and your capability, okay? Suddenly I have to become a salesman, all kinds of stuff like this. And right now, I was going to take the last bit of this. How much time do you have? 10 minutes or so?
That's pretty close.
Yeah, okay. I'd like to take the last bit and go fairly quickly in the how I got to where I am today, okay? You started it, so I'm going to finish it. How do you transition from being a nuclear engineer and physicist into, why isn't this showing up? Over here. Oh. Close caching. Okay, hold on.
Is it on the tablet?
It's not on the tablet.
Okay. That's a problem.
Okay. Screen sharing. So let's kill this and we'll restart it. Okay, there's now it woke up. All right, good. Okay. All right. So how do you get from being a raw nuclear engineer to suddenly being in medicine? Okay. Well, as was mentioned, I went to Rolla and then I went to Madison for grad school. Well, this is what I've done. All right. Grad school, well, here I had summer jobs while I was at Rolla to make money. My parents, we grew up in Louisiana. My first job was, I was 11, I was working on sugarcane, getting sugarcane at 11, and I have all my fingers and toes. It's really amazing. So I had to work. Okay, so I worked, had summer jobs. I worked at KMNR. I was a volunteer in Tom Dillon's research lab. He's the guy who got me interested. He's a professor way before y'all, but who got me interested in fusion. In grad school, I did research. I also was a consultant on a legal case. That really was a case of how to make a hydrogen bomb that a magazine wanted published, and the physics department was asked to provide researchers. So I worked on that case for 1.5 years. I got a very high level, top secret nuclear weapons grade security clearance in 3 days because the judge said, we're going to throw the case out unless you do it. So it was unheard of. I'd had security clearance for that, but not like this. They had to do it. And then I worked on that case. And then you can read about it. It's the Progressive magazine versus the Department of Energy. Eventually, somebody else published another paper, but basically what we showed, my job was to do research to provide evidence to both sides. And the evidence was, hey, is there enough information out there in the libraries of Los Alamos and places like that for someone to build a thermonuclear weapon? And the answer is, you know, you still need a little bit of nuclear material. Okay? By the way, I can teach a grad student how to make a biological weapon in a week. And we can order all the stuff we need, and it's really easy, and it's really whatever, and it's dangerous. So I'm not worried about nuclear stuff anymore. It's the biology stuff I worry about. Okay. All right, I went to work at General Atomics straight out of graduate school. I did work mainly in Japan, as I said, And then I started working on this human genome project and got hooked up with this guy. We then went to UT Southwestern in Dallas. I had an endowed chair, did bioinformatics and genetics. I suddenly switched to supercomputing. I got the very first supercomputer at that campus. I got Hewlett Packard to give us one. And then we started analyzing data. During that time, I founded a couple of companies that were eventually sold to Roche and other places. Then I went to Virginia Tech. I took over this Bioinformatics Institute, had 240 full-time employees. In my first 2 years there, I brought in $82 million, and we drove it to 340 people. Okay, and then the vice president for research came after me, which is why this is 4 + 4. And I say, just go and Google my name in Virginia Tech. Okay, I left there at year 4, but then they gave me an appointment for 4 more years to just pay me to keep my mouth shut. But I started working at this company, at this private medical school. By the way, the guy who was the head of the committee that oversees the whole university founded this school. So the day after this all got settled, he hired me there. Okay, so to help drive the research at this medical school. We then went from 1 campus to 4 campuses: Virginia, South Carolina, Alabama, and Louisiana. I helped build all of those. These are all running, cranking out medical students at about 170 per campus per year. They all do research, and so during that time, I did other research that helped drive research here, but I continued this microsatellite stuff, and I founded the company Orbit Genomics while I was there. So now I'm trying to slow down a little bit and not have so many jobs. So now I have this job, and I do consultant for a new medical school in Chicago. And also I have this farm called darkmatterhabitat.com, where we do rescue animals. And we actually now rescue people too. We have 3 people that live up above our barn. So that's what we do. All right, really quickly, this is General Atomics. If you've never seen it, it's not a bad place to work. It's right, it's a short run over to the ocean. Okay, I worked on fusion machines, the GA tokamak and other tokamaks. I worked in Japan on 2 different machines, one in Nagoya, Japan, one in Tsukuba. These are some mirror machines, mirror fusion devices. The last 6 years I was at General Atomics, I worked on all kinds of strange things, including biology. I worked on chemical demilitarization. How do you get rid of chemical biological weapons? Anybody recognize this? Yeah, so I worked on the 0th version of that before we made it, put it together. It didn't look like that first. So what was unique about that was it was the first UAV that Israelis had huge numbers of UAVs and put a camera on it and a person was there to fly it. This was the first one to put a GPS on it, so you didn't have to fly it. All you had to do was push a button, say take off, go over there 400 miles and go around and around in circles, or take a picture, or ram into that building, or later on shoot Hellfire missiles. My job was develop alternate mission scenarios for it. All I can say is everything I came up with, they do. So worked on railguns and a few other odds and ends and submarine communications. It was a fun time. Made a new kind of particle accelerator out of a microwave oven. That was rather fun. And it's a real easy way to make lots of x-rays. Okay, you put that thing in a vacuum with a little bit of a magnetic field. And so it has no Ds. You've seen these things where you reduce and accelerate particles because there's Ds and they alternate. If you put a microwave field in here that's locked to this bar, it will, and it's the 0-0 mode, it will alternate, creating the effect of Ds without having Ds. So it's the first D-less particle accelerator made out of your common household microwave oven that makes a boatload of radiation. Worked on the paints for these things. I didn't actually work on the paints. I worked on the question came down from the defense group that said, hey, what happens to stealth paint right here? Where the engine nozzle is, and it gets really hot. So I ended up building a device where I shot a CO2 laser in this little port, had a sample in there, and then I could heat it up. And it was in a chamber, and I could sweep it from 0 to 36 gigahertz, covering most radar ranges. And the answer is, the hotter it gets, the better it is. So, that, so I didn't know that, by the way. I started working on that project because someone asked, hey, could we do this? And I said, I think we can. I can't theoretically calculate it, but I think I can measure it. So my boss goes, well, here's some money, go and do that. 2 years later was when the first thing fell out of the sky in California. They had to admit there were stealth planes. I was working on it. I didn't know it. I didn't know I was working on this. Often happens with classified work. You didn't know what you're working on. So 2 years later, when one fell out of the sky and General Atomics had a big celebration, lots of Coors beer and stuff like that out in Milan, they said, we work on avionics for stealth planes and other things, blah, blah. I went over to the guy in the defense group and I said, was I working on that? And he goes, I can neither affirm nor deny. I go, okay, I got it. So, yeah. Worked on other things too. How many people have blue topaz jewelry? Well, you got some. Okay. All right. Do you know that most blue topaz jewelry is made by General Atomics? Nope. Most topaz is mined as clear topaz in Sri Lanka. It then goes to Hong Kong, where it's cut into stones, and then it's clear. It's worthless. They send it to General Atomics in 55-gallon drums. They drop it in our nuclear reactors or particle accelerators. I worked on what's called color center reactions in here. And so how do you make them different color blues? And so you run them in a reactor for a while, then you set the cans out. Well, you put them back in cans and set them out, let them sit for a couple of years, let the radiation die. And then you ship them to Germany as modified gemstones, not saying how you modified them. Then in Germany, they unpack them and then they sell them to the world. Okay, so in other words, General Atomics has lots of problems. This is where I started doing DNA stuff while I was working here. I invented this little thing. There's capillaries. We started selling those. And I made a thing that extracts DNA from cells, named it Helix Prepper. Unfortunately, this company in Texas threatened to sue me and everybody because they thought Prepper sounds too much like, because we called it Dr. Prepper. And so we actually ended up saying, okay, we'll change it to something else. But we said, if someone is going to buy for 50 cents a refreshment, they're not going to confuse it with something that costs $100,000 and extract DNA. But it doesn't work with lawyers at all. I went to UT Southwestern. I made more robots. I made DNA synthesizers, which is made this other company that then Didi went. Made more things to automate the whole DNA process. That's part of the Genome Center. And I'll end with this. One of the other things is my endowed chair came from Texas Instruments. And so Texas Instruments made this thing called digital light processing. How many people have a DLP TV or DLP projectors? They're basically little micro mirrors that are on a chip like this. And so I was encouraged to do things with TI technology. So I said, give me some. And this was before they came out as projectors. I figured out that if you take a projector with a is that a DLP projector? I don't think so. Generally, you can see. Maybe. I took the light out and replaced it with a UV lamp. Okay. And then you can project UV light. If you project UV light, you can do chemistry. I know. Okay. All right. Bottom line was, I did all kinds of stuff. I made these arrays. This became something called Nimblegen, which then wrote it down. I also, here's the last thing. I made holographic TV out of these things by putting in the holographic transform onto the chip and then projected it into different layers. Bottom line was I got an unlimited amount of money to make this into the first holographic TV, which the university promptly rejected and said I could not take that money. The reason why is the money was offered to me from investors in Las Vegas for the adult entertainment industry. You really like to have good 3D adult entertainment. So we ended up not making our first holographic TV because of the university rejected. So that brings me to today. And I'm here today to hang around with y'all. So thanks a lot.